Hello everyone, so for this video I will be showing you how to identify the different types of epithelial tissue when you're looking at them under the microscope. So first off, what is epithelial tissue? Epithelial tissue is tissue that lines various body surfaces and cavities, and it will be resting on a basement membrane of connective tissue, and it will also have this harsh line that you can see, and either above or below that harsh line, depending on how you're looking at it, there will be empty space. That is one of the key things to look for when you're trying to identify if it is epithelial tissue or some other kind of tissue. Next, we'll look at the different types of cells seen in epithelial tissue. And once you learn the cell types, it will be easier to identify what type of epithelial tissue you're looking at under the microscope. First, we have simple squamous, and whenever you see one layer of cells, then you know it's going to be simple. Simple just means one layer of cells. And squamous is referring to the shape of the cell. Squamous cells are flat and they look like they're squished down into scale looking like cells. And next after that is simple cuboidal, simple one layer, and cuboidal, as you might have guessed, just means it's in the shape of a cube. Columnar, simple columnar is just in the shape of a column. And when you get into multiple layers of cells, that's when you would call it stratified. And the last type of cell that you would see is pseudostratified columnar cells. Pseudostratified meaning that it looks stratified, like the tops of the cells are at different levels, but it's actually still just one layer of cells. Here's a picture of the alveoli under the microscope, and the alveoli are the tiny air sacs in your lungs where gas exchange happens, and this is a place in the body where you would find the simple squamous epithelial tissue. And it's kind of hard to tell, but there is only one layer of cells, and that's true with all alveoli, and they're very small, flat-looking cells compared to what we're about to see. So here we have a picture of the kidney tubules under the microscope, and we're looking at the two large circles near the center of the picture, and they are going to be lined with the simple cuboidal epithelial tissue. And this one's not too hard to figure out. There's just one layer of cells, so it's simple, and the nuclei line up pretty nicely, and it's just a cube-shaped cell, and that's how you tell if it's simple cuboidal epithelial tissue. So this is the slide of the gallbladder, and the gallbladder is lined with the simple columnar epithelial tissue. You can see that these cells are much more stretched out than the cuboidal cells. And also something I forgot to mention with cuboidal cells, the nucleus will usually mostly be a circle. And in here, if you can't tell what you're looking at, look for a stretched out nucleus that looks more oval shaped than the circle that you saw in the cuboidal cells. So simple one layer of cells, columnar, epithelial. Remember, you can see that harsh line there and the empty space past it. So here we are looking at a picture of the trachea, and the trachea is lined with the pseudostratified columnar epithelial tissue. And you can tell that it's pseudostratified because not all of the cells are at the same level. Some are longer than others, some are shorter than others. And this type of cell in the trachea is ciliated. It has cilia at the top. That's what you're seeing there. Some places of the body where you see this type of tissue will not be ciliated, but in the trachea, it is ciliated. The next slide is the skin, and at the top layer of the skin, we are looking at stratified, squamous epithelial tissue that is keratinized. So keratinized means that very top layer is dead. Underneath that top layer, there are living cells, but when you're looking for stratified squamous epithelial that's keratinized, you're only going to be identifying that top layer of cells. And if you look at it, underneath that top layer, you can see a bunch of nuclei, the darker spots in the cells, and keratinized tissue will not have nuclei anymore. So that's one of the things to look for when you're trying to identify the keratinized cells. They will not have nuclei. Here we have a slide of the esophagus, which is also lined with stratified squamous epithelium, but this is non-keratinized. You can see all the nuclei in there, those dark little spots in the cells, and it has multiple layers down to that dark basement membrane, and so you know that it's stratified, and the cells are that flat scale-like shape, so you know it's squamous. And again, the harsh line, you know it's epithelium. Stratified squamous epithelium that is non-keratinized. They're living cells in the esophagus. So this next slide is a sweat gland, and in that tubule there in the middle, we are seeing that it's lined with multiple layers of cells, so it's stratified. And the nuclei in there are somewhat circular. I know it's kind of hard to tell with some of them. They look stretched out, but the cells are cuboidal cells, and so we have stratified cuboidal epithelial tissue lining the tubules within the sweat glands. So this next slide is the uterine gland, and in the uterine gland, as you can see, it's stratified columnar epithelial tissue, and you're going to look for 2 layers of overlapping cells, and also look for an elongated shape and an elongated nucleus. And that's it. That's the stratified columnar epithelial tissue within the uterine gland. This last slide is a picture of the urinary bladder. And the urinary bladder is lined with a special type of tissue called transitional epithelium. And these cells have the ability to change their shape. And what you're looking at now is an empty bladder. These cells in the empty bladder will look more cuboidal in shape, and that top layer will have dome-shaped cells. As the bladder becomes more full, the cells will transition into a more squamous shape to create a bigger space for the urine so it can fill up. Thank you all for watching the video, and if there's anything else you'd like to see, please let me know in the comments.